Good day. My name is Carlos Bustamante, and today we are going to talk about the use of single molecule methods, specifically single molecule manipulation methods in biochemistry. In the first half, we are going to talk about why do we want to use single molecules and some of the rationale of why do we actually study these reactions at the single molecule level, what instrumentation is used to do that kind of experiments. And in the second half, I'm going to present you a particular application of these methods of single molecule manipulation to study a molecular motor. I want to start by reminding you that most of chemistry and biochemistry has been done by using the ensemble approach. In the ensemble approach, essentially you can follow a chemical reaction between a component A and a component B to produce a component C, as indicated here in this reaction. And the idea is that you mix a very large number of molecules of A with a very large number of molecules of B to produce also a very large number of molecules of C. In fact, I indicate here that typically we work with the order of Avogadro number of molecules, 10 to the 24 molecules. And I want to, in the first movie here, you will see that in fact, that's essentially what we do when we do a titration, for example, in the lab, in Chemistry 1. You had a compound with a particular color at the beginning, you add a second compound, and as a result of the reaction, the mixture actually changes color. And again, you add yet another reactant, and then again, the mixture changes color once again, indicating that the reaction is proceeding. Essentially, you are following the reaction by the change of this global ensemble property of the solution. Now, this is what we call the ensemble approach, and also is called bulk methods of studying chemical reaction. And in this particular talk, we are going to refer to them as well as studies made in multiplo to indicate that we use many molecules to actually follow the reaction. Now, there are many advantages to this way of studying reactions in bulk. And it is first that it is a very robust method because the signals are proportional to the number of molecules. And we've seen that actually there are lots of molecules in reacting A plus B to produce C. On the other hand, we must remember that what we are doing is we are following, in this case, what happens to the population on average. See, in this case, we are following that by the change in one property. In our particular example, in the movie that you just saw, that property is the color of the solution, and we see how that color of the solution changes over time, and that change is essentially a change that we are following over the whole population of molecules. Now, in reality, of course, if we had a microscope that would allow us to enter the solution and see the individual molecules, we will find a completely different picture. Every molecule will be doing something very different. For example, they will be reacting some very fast, some others will be reacting at some intermediate rate, and some will be reacting very, very slow. And so still, what we follow, of course, is the average of these behaviors, these molecular behaviors. And yet, we can learn quite a bit about the reaction. In fact, there are, however, limitations to this approach, right? Because despite their power and their robustness, we actually have to compromise quite a bit in order to study a chemical reaction in bulk, as an ensemble, in multiple. And is that, in reality, we are not following the molecular trajectories, the trajectories of the individual molecules as they carry out the reaction. Instead, what we are actually doing is we are following the dynamics of the mean of the population, something that we actually call, in chemistry, kinetics. Okay? Now, in reality, what we would like to do is to be able to describe and to see how the different molecules in the solution are actually carrying out the reaction. But that requires that instead of studying the system, the molecules, the reaction, as an ensemble of molecules, that we actually follow the reaction one molecule at a time. So you may ask, why do we want to follow reactions one molecule at a time? Are there any advantages in doing that relative to the ensemble or bulk approach? Well, I would like to point to you that one place in which following reactions of very few number of molecules or following reactions at the single molecule level is important is the interior of the cell. In fact, many cellular processes we know now, such as, for example, chromosome segregation, chromosome replication, transcription of DNA, translation, recombination, are often carried out by very few molecules in the cell. So here is an interesting back-of-the-envelope calculation. What you see here is a cell of E. coli, and you may ask, given the volume of an E. coli cell, imagine that you have a single molecule of any kind, indicated here in this yellow square, inside this E. coli cell. And now you ask, what is the concentration of that single molecule of that particular type inside that E. coli cell? Because the volume of the E. coli cell is so small, it turns out that one molecule of E. coli in an E. coli cell is at a concentration already of 1.6 nanomolar, right? And that is a very interesting result because it suggests that many reactions can occur inside the cell with just having one single molecule of any kind inside the bacterium, right? In fact, imagine that that protein is a DNA binding protein. We know that the affinity of most DNA binding proteins is below nanomolar. Therefore, the presence of one single molecule of that protein in the cell, giving a concentration of 1.6 nanomolar, ensures that that protein will be 90 or 99% of the time bound to its target site in DNA. In other words, reactions in the cell, because of the tiny volume of the cell, can occur when the molecules are present at very, very small numbers. Very, very small numbers. So, why do we want to study single molecule or reactions at the level of single molecules? Well, there are many questions that we may want to answer if we are able to follow reactions at the single molecule level. For example, first, how do reactions in the cell occur when you look at them one molecule at a time? What are they telling us about how the mechanism of the reaction is, what the mechanism is? We may want to also find out whether all the molecules follow the same trajectory every time you go from A to B, or whether they follow different trajectories or different paths. We also want to know, perhaps, how noisy are the processes in which molecules are reacting one molecule at a time. Remember, now we are looking at very few molecules, and you expect that there could be great fluctuations in the behavior of these molecules and in the signal. And can we learn something about these fluctuations, or this is just noise that contains no information? So all of these questions are interesting questions that become accessible if we develop methods that would allow us to study chemical reactions one molecule at a time. Of course, we must be prepared to pay some price for studying single molecules. These studies, which we can actually call in singulo, to indicate that we are now studying a reaction one molecule at a time, have signals that are very different from the bulk signals that, remember, were very robust, very quiet, very nice. In this case, your molecules will instead display a very fast instantaneous dynamics, which means that the behavior, the signal that you measure, the behavior of the molecules, actually may appear random or sometimes stochastic, in other words, very noisy. You will see that fluctuations in this case dominate, in a sense, the magnitudes of the signals, and you may find that the molecules may be found sometimes to coexist in more than one state. Sometimes they are in one state, sometimes they may actually go into a second state or a third state, and they may be actually moving along this path, coexisting in these different states. And even in some cases, molecules may be in very unlikely states, but then all of a sudden they enter these unlikely states, and you may observe them doing that. This is what we call extreme states of the molecules. But they are part of the behavior of the molecule, and as such, they are of interest, of course, for studying. I want to illustrate some of the advantages of studying single molecules by pointing out to you that, as I mentioned before, molecules sometimes may not exist just in one single state, but they may coexist in several states at equilibrium, right? And so, in fact, the molecular property that you are interested in studying, for example, let's assume that is the ability to absorb light, or the dimensions of the molecule, or, for example, the state of the molecule, whether it is as a monomer, or if it is as a dimer, forming a dimer, or aggregated in some larger structure, whatever the molecular property that you are studying may not be actually a single population of them, but actually you may have a multiple population where you may have molecules spending some time in this type of property with this particular value of the property, or they may actually spend some time also in this other property, and that there may be some kind of exchange between these two populations, right? If you are using an ensemble method, of course, what you are going to get is not information about the existence of this population or that population, but typically what you will get is actually the average of the two populations. That's the signal that you are going to measure, and the molecular property that you are going to actually be studying, in this case, is going to be the value given by this green bar, as indicated here. But it is also possible that the molecules actually exist in a single population that is very broad, as indicated here in this second panel, right? In this case, you may end up actually having the same average, but now you have just a single population that has a rather broad distribution. Or yet, in even more extreme case, you may have just a single population, as indicated here, but with the same value of average. So notice, in fact, that in these three panels, the one on the left, the one in the center, the one on the right, actually a bulk method, an ensemble method, will give you the same answer, even though the behavior of these molecules is very different, because in one case you have a multimodal population, whereas in the other two cases you have actually a single population. Okay, so what I would like to do is now motivate the study of single molecules by telling you a little bit about how, in my laboratory, we started studying molecules one at a time. And for that, I'm going to tell you a story about the elasticity of single DNA molecules. Many years ago, we were studying how molecules of DNA actually move inside a gel. As you know, electrophoresis is a very powerful technique that biochemists constantly use to separate molecules of different size, molecules of DNA of different size, or molecules of protein molecules of different size. In this particular case, we wanted to know how is really that molecules of DNA separate by size. In order to do that, what we did was an experiment where we mixed molecules of DNA with ethidium bromide, which is a dye that intercalates in the DNA and that renders the molecules fluorescent. Now, we mix these molecules of DNA with a gel, in this case was agarose gel, and then we put them between a slide and a cover slip, allow the gel to harden, and then we seal this cover slip with nail polish. It is always good to actually have some nail polish around in the laboratory. And then we applied 2 electrodes on both sides of the slide, and we look at this slide cover slip type of chamber under a fluorescence microscope, an optical fluorescence microscope. And what you see is, what we saw, is what you see in this movie. As you can see, actually the molecules of DNA, in this case these are molecules of T4, bacteriophage T4, are moving all towards the positive electrode, right? But notice that the molecules are actually extending as they go to the positive electrode, both arms in the direction of the positive electrode, but one of the arms is usually larger, just by chance, than the other, has more charge, more phosphates, and therefore experiences more force, and it will win the battle, and will end up actually pulling the other arm around the corner, as indicated here, right? There it goes. Notice how it pulls around the corner, and the molecule actually collapses again to start a new cycle. This repeats over and over, but what I would like you to notice is how, when the collapse occurs, how elastic the molecule appears when the collapse occurs, right? In other words, when that particular arm turns the corner, notice it catches up very fast. And that really surprised us quite a bit. We, in fact, knew about elasticity, but seeing the elasticity at play actually was a great motivation. And so we thought, why not actually trying to understand the elasticity of a single molecule of DNA? So we decided to actually do an experiment to study the elasticity of a single molecule of DNA. And the idea is quite simple. As you see here in the slide, the idea is to attach one end of the DNA to a wall, right? And then grab the other end of the DNA and simply ask, how much force do I have to apply in order to extend the molecule a given amount? That's essentially to try to understand what the elasticity of the molecule is. Of course, in practice, the way you do this experiment is by attaching the DNA to two microscopic beads, in this case, plastic beads. One of the beads is kept atop a micropipette by suction, as indicated here, right? In that one, the second bead. And the other bead, which is the one that you are going to apply the force to, actually is attached to the other end of the DNA. And so the idea is then, again, we pull on this second bead out, and we determine how much force we have to apply in order to extend the molecule by a given amount. Now, how are we going to apply the force? There are many ways in which we can apply the force. We could, for example, use a magnetic bead for the second bead, for this second bead, and then bring a magnet near the microscope, and in that way apply a magnetic force that will stretch the DNA. Alternatively, we can actually use what is called an optical tweezers instrument. Optical tweezers were invented in 1970, and they are the result of an observation made by Arthur Ashkin many years ago, where he found that because light carries momentum, it has the ability to trap objects that have a different index of refraction than the surrounding medium. And so you have a beam intensity, as shown here, that is typically Gaussian for a laser, okay? So the light is more intense at the center than it is at the periphery of the beam. Then if you approach a bead close to the beam, what will happen is that that bead will experience a net force, as indicated here, that will tend to suck up the bead towards the center of the beam. Once that happens, then every time you move the beam, the bead is going to be essentially, the bead is going to follow the beam everywhere. The bead is basically trapped in the beam of light. And that's a great thing, because you can actually now use light to manipulate things far away. In other words, you can manipulate these microscopic beads, as I showed you before, and if those microscopic beads happen to be attached to a molecule of DNA, now you can use those beads and the beam of light to manipulate your molecules of DNA. So, in fact, in the next slide, you can see how do we go about attaching the molecule of DNA between the beads, because obviously that's a condition to do this experiment. You see on top 4 panels, which I have labeled A, B, C, and D. So the idea is the following. What you do is you grab a molecule of DNA and you modify the ends of the molecule of DNA, placing a biotin at both ends in this case. Then you have beads that are covered with streptavidin, which bind very, very tightly to biotin, as you know. And then we place that bead atop a micropipette, as indicated in panel A. And then we pass molecules of DNA by flow through the chamber, both of ends of which are actually labeled with biotin, remember? So that's indicated here in the A panel, and the arrows indicate here that we are flowing the molecules of DNA, as you can see here, right? Now, just by chance, we expect that one of the molecules of DNA will actually hit this bead, and it will react, the biotin of one end with the streptavidin on the surface. Because we are applying flow, that molecule of DNA is going to be extended, as shown here in panel A, right? As you can see here. Okay, now that you have the molecule hopefully attached, now you pass more beads and you turn on the optical trap. The optical trap or optical tweezers is indicated here by the tick marks, the 4 tick marks. One of the beads will pass by the trap and will get trapped in the optical tweezers trap, right? In the center there. And now, what you do is, still in the presence of the flow, because you want to maintain the molecule of DNA extended, and knowing that that end of the DNA also has a biotin that can react with another streptavidin on the surface of that bead, you then now approach, by moving the pipette, the bead in the trap to the bead in the pipette, encouraging the formation of a single bond, as indicated here, between the biotin at this end of the DNA and the bead. And when that happens, you should be able to actually pull the pipette and, in fact, fish the bead out of the trap using the DNA as your fishing line, right? Okay, so that's the idea of this experiment. And you will see in the movie here that we are already in panel B, because we have a bead in the trap and a bead in the pipette. Hopefully we have a molecule of DNA between the two, but the binding between the second biotin and the streptavidin hasn't occurred yet, but we are hoping that it will occur. And in fact, there, you can see that immediately that it happened. So now you see that, in fact, we have now a molecule of DNA between these two beads. And the process is so efficient in a sense that notice that actually we also got another molecule of DNA between that bead and another bead that was in the trap. So in fact, we have bead DNA, bead DNA, bead, right? So it's a very efficient process, and it only took a few seconds to actually put a molecule of DNA between two beads. Okay, having the possibility of doing this allows us then to really pull the beads apart and study how much force do I have to do in order to apply in order to extend the molecule a given amount. And the result is shown here. This is what is called force extension curves of DNA. And you can see that in the ordinate, I am actually plotting here the force required to stretch the molecule, and in the x-axis or the abscissa, I actually have here the extension, the relative extension of the molecule of DNA. And you see that the molecule of DNA does behave as a spring, but it's a very non-linear spring. Notice that the data here actually curves in a very, very, very sharp way, right? Indicating that the molecule, in fact, is not a linear spring. If the molecule would behave as a linear spring, it should have, in a plot of force versus extension, as this is, right? This plot is force versus extension. We would expect the molecule to behave as the dashed line here in the middle, right? You see that actually that's what would be a Hookean spring, a linear spring. DNA is not a linear spring, it's a highly not linear spring. Okay, so in fact, we studied this for quite a while, and we were able to show that it is possible to rationalize all this elastic behavior of the DNA molecule using a model of polymer elasticity called the worm-like chain model, and we derived this expression shown here that actually accounts very well for the force needed to extend the molecule a given amount, in this case z, when the molecule has a length, a contour length L, and the experiment is carried out at the temperature T. This particular model requires the introduction of one parameter that describes the elasticity of the DNA molecule, and that parameter is what we call persistence length. This interpolation formula, which I show here in this panel, actually turns out to be a very, very good approximation to the exact solution for the force extension behavior of a molecule of DNA. But you may ask, how do we go from pulling a molecule of DNA to actually study and following reactions in singulo? It turns out that the ability of manipulating one molecule of DNA, as I show you here, allows us to follow the activity of a number of molecular machines that are actually nucleic acid-based molecular machines. And so I want to then tell you a little bit about these molecular machines. Many years ago, some 25 years ago, biologists still thought of the cell as a small bag of a concentrated solution of nucleic acids and proteins that were undergoing second-order reactions. But for the last 25 years or so, there has been a paradigmatic shift in the way biologists look at cells. Now, in the words of Bruce Albert, in fact, in this quite celebrated paper in Cell in 1998, he pointed out that the entire cell really is better described as a factory that contains an elaborate network of interlocking assembly lines, each of which is composed of a set of large protein machines. And so the idea, the paradigm that has emerged in the last 20 or 25 years or so is that the cell, in fact, has a modular factory, a modular architecture. Essentially, each central function of the cell, and I have labeled here, mentioned several, replication, transcription, translation, translocation, recombination, splicing, protein folding, protein degradation, is really performed by specialized multiprotein assemblies. And these processing units actually typically function as molecular machines. These molecular machines, here is one example, for example, the molecular machine responsible for the initiation of transcription in eukaryotic cells, which has more than 60 proteins involved, actually, are responsible for one particular function. And they contain various parts with specialized functions. They have dimensions that are in the nanometer scale because they are very, very large. And often they provide a localized environment for reactions to occur, right? So, in fact, many of them function as molecular motors, converting chemical energy, which could be in the form of ATP hydrolysis or GTP hydrolysis, or in the form of a gradient of protons across a membrane, into mechanical work, producing movement and the generation of force. Well, it turns out that single molecule manipulation methods, as I showed you before, are ideally suited to study these molecular machines, and particularly these molecular motors, because force and displacement, which are the two variables that are most natural for the behavior of these motors, actually are direct observable in single molecule manipulation experiments. So, in fact, we can now follow a reaction such as this. A single molecule of RNA polymerase, as shown here, bound to the promoter and transcribing a single molecule of RNA and synthesizing a single molecule of mRNA. And we can actually follow that reaction in real time, one molecule at a time. How do we do that? In this case, we will use two optical traps, as indicated here, one trapping one bead that has attached by the back a single molecule of RNA polymerase. This is the eukaryotic RNA polymerase Pol2, and that molecule is bound already to its promoter, and so you have the rest of the DNA, and the other end of the DNA is attached by a biotin or a digoxygenin or some other chemical moiety to a bead, which is also kept in a second trap. And now we can in fact play tug of war with one of these molecules of RNA polymerase. As we add the NTPs, the nucleoside triphosphates, the molecule will want to actually transcribe the DNA. And in order to transcribe the DNA, it has to, because it cannot move, because it's attached to the back, by the back to this bead, in the laboratory frame, it has to pull the DNA through itself. And in pulling the DNA through itself, it must pull this second bead out of the trap. and we can follow that movement of the second bead out of the trap, and in this way follow the chemical reaction, the transcription of a single molecule of RNA polymerase in real time, one molecule at a time. And in fact, you can see here that because the distance between the beads is getting shorter as the molecule transcribes, you can see that, or larger if you actually do the experiment in the opposite geometry, then you can actually follow how the length or the position of the bead changes in the trap as the time passes, right? So notice that in this case, the polymerase is not behaving in a very simple manner. For a while, it moves, which corresponds to the regions here in the curve that are black, but then for no apparent reason, the molecule stops. It has a pause. It pauses there for a while, and again, it resumes transcription for no apparent reason a few seconds later, and so on and so forth. This kind of dynamics is not accessible in bulk experiments, and it only becomes accessible when you are able to study molecules one at a time. Here is another experiment where we have changed slightly. We are still studying transcription one molecule at a time, but what we've done is we have now a molecule of DNA attached between two beads, as indicated here in this slide, and one of the beads is kept in an optical trap. The other bead is, as before, atop a micropipette, right? And what we have now is a molecule of RNA polymerase to which we have attached a tiny little bead on the side, and in order for the molecule to actually transcribe the DNA, it must go around and move around the DNA, the spiral of the helix of DNA, and we will be able to see that in the movie because the bead is large enough that allows us to follow the actual movement around the DNA as the RNA polymerase transcribes the DNA, as you can see there. Again, this type of experiments allow you to follow the dynamics of the reaction, not just the average behavior of RNA polymerase molecules, but how individual molecules behave one time and the following time and the following second and so on and so forth. Another experiment that we can do, we can actually follow a whole ribosome. As you know, this is a very complex machine made out of more than 50 proteins and two different types of DNA, of RNA, but we can actually follow this process in real time, right? And so in this case, the experiment is different. We put the ribosome at the base of this RNA hairpin, which is going to be translated by the ribosome, and then we have grabbed the two ends of the RNA hairpin, and we have one attached to a bead in the pipette, and the other attached to the bead in the trap as before. So now what is going to happen is this. We add all the initiation factors, the tRNAs, and the elongation factors, and then this ribosome is going to start translating this RNA. As translation occurs, the ribosome has to open the hairpin here, right? And as the hairpin opens and is being translated, essentially the molecule, the RNA molecule, the arms of the RNA molecule are getting longer and longer, which means that the beads distance actually gets separated, right? So we can actually follow the reaction, in this case, by the beads moving apart as a function of time as the ribosome actually enters the hairpin and opens the hairpin. And what you will see here is, in fact, translation by a single ribosome. The dashed lines in this plot are separated by 3 base pairs, and perhaps not surprisingly, but remember this is one single ribosome translating one single molecule of RNA. What we see is that the ribosome is in fact moving by one codon at a time. 3 base pairs, 3 base pairs, 3 base pairs every time. Note that at the end, actually, there is a big jump, and that big jump is because what is left of the hairpin is so small that it's quite unstable at the tension that we are applying at the ends of the RNA, and the RNA just spontaneously opens. Okay, so we can also study the ribosome as a molecular motor, because obviously the ribosome, in order to move along the mRNA, has to be converting part of the chemical energy of the reaction that is catalyzing the peptidyl transfer reaction and the peptide synthesis into mechanical work and force. And so in this case, what we do is we grab the ribosome by the back, as before, and attach it via a biotin and a streptavidin linker to the surface of the bead in the pipette, and the other end of the mRNA is grabbed by a piece of DNA, forming a DNA-RNA hybrid onto the surface of a bead that is in the trap. And now this is what we call our tug of war geometry. And now when we add all the reactions for translation to occur, or the reaction mixture for the translation reaction to occur, what we'll see is that the ribosome, in order to translate this mRNA, will have to pull the bead out of the trap in order to actually move along the mRNA. And in that case, you can see, as shown in this movie, you can see that, in fact, again, notice that the dashed lines are separated excuse me, by 3 base pairs, and you see that actually the ribosome strictly moves along the mRNA by a codon at a time. And we can follow that in real time. And you can, more importantly, notice that the ribosome spends a time at each codon before it actually makes the next translocation. How is this time distributed? What is happening during these times? How sharp the distribution of times, these waiting times are? How fast the actual translation from one codon to the next codon occurs? How does that depend on the force applied or the tension applied to the RNA? All of these are questions that we can now access because we have a single molecule assay for this experiment. And in fact, in this experiment, just to finish this first half, I like to show you that we can apply increasing tension in this tug of war experiment between the ribosome and the other end of the mRNA, and see how the rate of translation of the ribosome varies with the applied tension, with the load. In other words, we can study the ribosome as a motor, and we are able to characterize for the first time how is that this motor, the ribosome, responds to a mechanical load. And here is the answer. You see that actually the rate of translation is quite sensitive to the force applied, and that the ribosome can work against forces around 14, 15 piconewtons, but then at that point, the rate of translation becomes very, very small. Okay, so this is an example or a group of examples of how you can use single molecule experiments to study complex biochemical reactions. In the second half of this lecture, I will now specifically talk about one particular motor that we have studied in great detail, and hopefully that will illustrate the kind of information that is possible now to obtain using methods of single molecule manipulation.